Dear student, in this video I am going to talk about electron microscope, especially transmission electron microscope. This is a kind of electron microscope. So before that, what is meant by electron microscope? Electron microscope has become indispensable in microbiological research because the high resolving power of the instruments that enables the visualization of the fine structure of bacteria, virus, cells and tissues, which is not possible with the light microscope. So electron microscope of the historical background, it was developed in 1930s and first applied to the biological specimen by Albert Cloud, Keith Potter and George Pallet in the year of 1940s and 1950s. The electron microscope can achieve a much greater resolution than the obtained with the light microscope. So the illumination source of electron microscope is a white hot tungsten filament which emits the electrons. The wavelength of the electron is shorter than that of the light. The wavelength of electron in an electron microscope can be short as 0.004 nanometer, about 1 lakh times shorter than the wavelength of visible light. So theoretically, this wavelength could be yield a resolution of 0.02 nanometer, but such a resolution cannot be obtained in practice because resolution is determined not only by the wavelength but also by the numerical aperture of the microscope lens. So numerical aperture is a limiting factor of the microscope. There are two types of the electron microscope, namely transmission and scanning. So first, let's see the transmission electron microscope, is shortly called as a TEM. In principle, viewing specimen under a transmission electron microscope is similar to the observation of stained cells with the bright field microscope. The specimen are fixed and stained with the salts of heavy metals which provide contrast by scattering electrons. A beam of the electrons is then passed through the specimen and focused to form image of fluorescent screen. So the electron that encounter a heavy metals ion as they pass through the sample are deflected and do not contribute to the final image. So stained areas of the specimen appear dark. The specimens to be examined by transmission electron microscopy can be prepared either positive or negative staining. In a positive staining, a tissue specimen are cut into a thin section and stained with heavy metal salt. That's heavy metal salts such as osmium tetroxide and lead citrate that react with the lipids, proteins and nucleic acids. So these heavy metals ions bind to a variety of the cell structure which consequently appear dark in the final image. So alternative positive staining procedure can be also used to identify the specific macromolecule within cells. So negative staining is useful for the visualization of intact biological structure such as bacteria, isolated subcellular organelles and macromolecules. So in this method, the biological specimen is deposited on a supporting film and a heavy metal stain is allowed to dry around its surface. The unstained specimen is then surrounded by a film of electron dense stains. A procedure and an image which is specimen appears a light against a stained dark background. What are the parts of the transmission electron microscope? First, the optical system and the component of spots. The transmission electron microscope and its optical system are: the electron microscope consists of a column of stack at the top of which is mounted the sources of illumination. The electron guns which emits the electron from the hot tungsten wire filament. So beneath the filament a cathode shield is placed. A high voltage which can be varied from 50 to 100 kilovolt is applied to the anode. So the life of the tungsten wires filaments limited and it's usually required by replace after 15 hours of viewing. A pencil beam of electrons moving at high velocity is projected through the hole in the anode and onwards down the stack. High accelerating voltage used must be stabilized to ensure uniform velocity of electrons. There is a condenser lens system which bend the rays of electron so that a parallel beam is directed into the object placed below it. The electron is scattered to a degree that is proportional to the thickness and density of various parts of the specimen. An adjective lens gathers the scattered electron through a variety of small aperture and bring them the focus where a real primary image is formed and is magnified about 100 times. Two projector lenses which have function of the eyepiece of the light microscope, magnifying the part of the primary image further 300 to 500 times. The focal length of the electromagnetic lens can be changed by varying the current flow through the lens and thus continuously variable magnification is obtained. The final image is observed on a fluorescent screen situated at the lower end of the stack and viewed through a glass window. So the screen can be withdrawn by lever to allow electron to impinge on a photography plate or film held in camera. So viewing the high resolving power of the electron microscope, it is possible to the negative at very high magnifications. So this is how about work the transmission electron microscope. Thank you.